Okay, so welcome, Duke alumni and friends, to this session of the Duke Alumni Forever Learning Institute, an interdisciplinary program built around a series of themed conversations and learning experiences. My name is Diontre Earl. I'm the Director of Development for Duke Science and Technology, which is in our Office of Alumni Engagement and Development. I'm honored to serve as your guest host and moderator for today's webinar. This is the second session of our Blue Devils in Space, part of our Duke Forever Learning Institute. Today's session is entitled Life Beyond Earth, where we'll explore how research at Duke and NASA informs our understanding of life in space, extreme environments, and human adaptation for long-duration space travel. We have three speakers today, each of which will have 5 minutes to present their expert spotlight. And after that, we'll move into a moderated discussion and then open it up for audience Q&A. So I'll encourage each of you as we go through to put your questions into the chat, and we'll get to as many of those as we can, as we want to cover as much as we can before the 1 o'clock hour. Now let's meet our speakers for today. Let's give a warm welcome to starting with Anna Breit. She's in our Duke Lemur Center. She is a research scientist at the Lemur Center, studying hibernation patterns of fat-tailed dwarf lemurs and how this research might inform human adaptation to long-duration spaceflight. Please welcome Anna.
Hi, everyone. I'm just gonna share my screen real quick. Yeah, so my name's Anna Breit, and I am the hibernation physiologist at the Duke Lemur Center. I study tropical hibernation, which is something not everybody has thought about. For me, it's very exciting because most of research has been conducted where it's cold, and so thinking about warm hibernation is kind of mind trippy. So yeah, I have experience traveling to tropical places and studying mammal body temperature regulation. And this is a fat-tailed dwarf lemur. I tried to squeeze in as many pictures of them as possible, but I also needed to put a lot of figures and graphs. So I'll try to sprinkle them in. On the left is a 10 rack, and I don't have time to talk about it, but maybe later. So traditionally, we think of hibernation as this, over the winter months, we have very cold temperatures, and you have this blue line is the body temperature, and so you have like a typical warm body temperature. And during this fattening pre-hibernation phase, the animals will drop their temperature in these little test drops. We call them like test bouts. And then you see the animal drop their body temperature to the temperature of the cave, of the den, wherever they're hibernating, for a few weeks or days at a time. And then they have these little spurts of warm body temperatures, and we call those interbout arousals. And animals can have a bunch or very few, but they do this regularly throughout the hibernation season until they warm up again in the spring. And these bouts are what they spend most of their energy on. So they're saving energy when their body temperature is really, really low, and their fat stores are being depleted by these interbout arousals. So they're very energetically expensive, which we would assume means that they're very, very physiologically important, but we're still trying to understand those interbout arousals. So traditional cold hibernation, think of like a bear in a bear den or bats in Canada. They're hypometabolic, so they have lower metabolism, and they're hyperthermic or hypothermic. Oh, yeah, hypothermic. That's a typo. Terrible typo. Oh my goodness. Hypothermic. So very cold temperatures where they're just keeping their body temperature at ambient temperature when that's normally very cold, right? And that happens during periods of low food and water availability. And so their cells are also very cooled down. Imagine putting, your food in the freezer. Not much is going to be able to grow in the freezer, right? So your cells, the cellular mechanisms are slowed down, like those homeostatic things. But it saves a lot of energy. So there's the trade-off that they're going through versus tropical hibernation. And again, this is a very young field. We're seeing that they're hypometabolic, so they're lowering their metabolism, but their body temperature isn't necessarily super cold. They're still maintaining, or not necessarily maintaining, but they're having warm body temperatures. And that's really interesting looking at like the cellular level of warm cells, but their metabolism is really slow. I'll get into that later. But that we think of, okay, but during winter, there's no snow on the ground. What are they doing that for? And it's normally during periods of low food availability, so like during the dry season. And because their temperatures are warmer, we hypothesize they'll have greater homeostatic control. So their cells are still warm, even if their metabolism is cold. But looking at the energetic savings, are they actually able to save the amount of energy that they need to during the low food availability? So try to put pictures to what I'm saying. On the left, we have the traditional cold hibernation. And so this dark blue line is ambient temperature, and this light blue line is what they're doing with their body temperature. So every once in a while, they're having these interbout arousals, these spikes of warm body temperature. And that we can look at the energetic cost of that strategy. So their metabolism is really low, it's almost free to keep your body temperature at ambient temperature. But once in a while, they make their body temperature get significantly warmer than ambient temperature, and that costs a lot of energy. Versus traditional tropical warm hibernation, we're seeing their body temperature fluctuate with ambient temperature. So their body temperature is almost the same as the ambient temperature, which should be relatively energetically cheap. So their metabolism stays relatively low. So they stay in that hypometabolic state. And the moment we've all been waiting for, these are the fat-tailed dwarf lemurs that I study at the Lemur Center. They are very unique. They are endemic to Madagascar. They store all their fat in their tails. So versus a really fat squirrel or chipmunk or bear, they can put their fat around their organs that are generating heat. If these animals were to do that in the tropics of Madagascar, they would overheat and die. So they store their fat in their tails so that they can still dissipate heat from their warm organs like their liver and stuff, so they don't overheat. So on the left is a picture of them, in their out and about. On the right is this is ostrich. He is my favorite lemur. We're not supposed to have favorites, but whatever. This is a video camera screenshot from ostrich in our hibernation setup. I like to show this picture because this is his tail. It doesn't look like a tail. This is how thick his tail is. And that's just at the base. The whole tail you can see very thick here. And this isn't even during their fattening phase. So they store huge amounts of energy as fat in their tails. And then they use that fat throughout the hibernation season. So our study, we compare cooler traditional hibernation to tropical hibernation, and these animals are able to do both in Madagascar. So in Madagascar, when animals are in a very well insulated tree roost, they're buffered from outside temperatures. So they're experiencing temperatures 20 degrees Celsius is right around room temperature, let's say 68 degrees. And this gray line, which is very hard to see, is the ambient temperature. And then this dark black line is their body temperature. And you can see that they're having these interbout arousals, but then staying relatively cool for days or weeks at a time, and then rewarming regularly. And then we have a second hibernaculum is where an animal hibernates. So we have a second room where we're controlling the temperature. And in that room, we're fluctuating the temperatures on a daily cycle. So we're going from around 12 degrees to 30 degrees. So we're going from like 56 to 85 degrees-ish Fahrenheit. And those animals in that room, we expect to allow their body temperature to fluctuate with the ambient temperatures. And that would mimic dwarf lemurs in Madagascar that are in a poorly insulated roost. So the daily temperature fluctuations are that extreme during the dry season in Madagascar. And if they're not in a well-insulated roost, they're experiencing those extreme fluctuations. So we're measuring that, and we're taking tissue samples at 4 key moments throughout the hibernation season. So we have the fattening phase while they're getting ready for hibernation, when they're doing those pre-hibernation torpor bouts, those little test drops. And then during hibernation, this is very tricky, but we try to take samples, and this is just like blood samples, little ear punches to grow skin cells later, nothing super invasive. So just, drawing blood. And it also serves as a checkup to make sure the animal is doing well. So we do the second test, the second tissue check is during the actual hibernation phase, when they're at those low points during a torpor bout, when they're hypometabolic and hypothermic. Then we do a second within hibernation season test at high temperatures. So during that interbout arousal, it's kind of hard to time when the animal is going to start their interbout arousal, but we try to time it. And then finally, after the hibernation season, when they're going back to their regular housing, we take a final measurement. So I specialize in respirometry. That's the study of breathing, measuring breathing so that we can infer VO2, which is a proxy for metabolic rate. And real quick, this is a rat that I studied in Borneo, but it'll serve as our model for the day. So we have the animal in a temperature control environment. And we know the amount of oxygen going into their environment is 20.94% oxygen. When they're inhaling oxygen, they're exhaling carbon dioxide, and that ratio changes. So they exhale less than 20.94% oxygen. Their carbon dioxide, let's say it was 400 parts per million. It's they're exhaling a lot more carbon dioxide. And then we also measured water vapor. So if they're panting, they're exhaling more water vapor. And we can look at that ratio, the delta between the incoming and incurrent and excurrent air. And from that, we know the flow rate. We know the percentage of incurrent and excurrent gases, and we can infer what their VO2 would be, what their oxygen consumption would be, and that tells us their energy expenditure. So I also can do this with cells. So we are taking little skin punches, like a 2 millimeter punch of skin, and then we're able to grow skin cells from that. And then we're trying to infer whole animal physiology from those skin cells. And so we have the cells growing, and you can do this with a number of different tissues. We currently only do anything with the little skin cells, and we're able to multiply those cells, plate them, and then measure the change in pH, which tells us the oxygen consumption rate. So very similar to what we're doing at the whole organism level, we can do at the cellular level as well. So here's just some preliminary data. The traditional cold hibernation room, the animals are following ambient temperature with their body temperature with these periodic spikes where they're raising their body temperature. And you can see that costs them energy. So this is VO2, this is oxygen consumption or the metabolic rate. And you can see it costs them energy every time they have these spikes versus in the fluctuating room, their body temperature is only a few degrees above ambient temperature or around ambient temperature. So their metabolism is actually, their VO2 is actually relatively low. So it looks like they're saving more energy. So we currently have a lot of projects going on looking at this. I'm looking at the transcriptomics, the lipidomics, so which proteins are being made, what's actually going on at the cellular level. We also have integrated some machine learning to look at presence, absence, and activity monitoring. So the animals in the fluctuating room that are able to rewarm are a lot more active, which is interesting. We're also developing customized collars so that we can look at their activity. We can also look at heart rate. Ideally, in the future, we'll be able to look at heart rate, breathing. With the blood that we're taking, we're able to look at inflammation and immune response. So a lot of hibernating animals, if they're infected with a pathogen, this isn't our research, but in other labs, when the animal is infected with a pathogen, they don't mount an immune response until those interbout arousals. So that could give us insight into what the purpose of those interbout arousals are. We're also looking at cognition. So before and after hibernation, we're doing these cognitive assays to look at memory and learning. And the idea was if we are to hibernate humans, when they wake up from months-long hibernation, will they still be able to fly the spacecraft, right? We don't want to wake them up and then have them be very drowsy for another few months and, take a left when they were supposed to go right, something like that. I'm just kidding. I know that's not how spacecraft works. But anyways, we're also looking at flat inflammation. This is where if you have fat deposits in certain parts of your body, so if you have a beer gut, it is unhealthy fat. It's more likely to be unhealthy, lead to problems later down the line versus fat, in the glutes and in the tail, presumably, is also not as detrimental. So these animals can put their fat in their tail. They can, choose air quotes, where they deposit their fats, which is really interesting from a biomedical perspective. Interestingly, hibernating animals, their telomeres do shorten, as you would expect during hibernation. But when they come out of hibernation, their telomeres elongate, and that's a proxy for aging. So hibernating animals typically live significantly longer than non-hibernating animals. So that would also be very interesting to look at. We have collaborators at the University of Washington and the University of Minnesota looking at that. And then we are hoping to look at the reproductive outcomes. So those individuals that hibernate regularly have been shown to have more reproductive success when they come out of hibernation during the mating season. So implications. Fat-tailed dwarf lemurs are the closest relative to humans that are able to hibernate. So they are primates. They have cognitive functioning, not similar to humans, but from the primate world, there are no other primates that can hibernate. They also will hibernate at warm temperatures, which if humans are to hibernate, again, we can fight on this. My line of thought is that humans should hibernate at warmer temperatures than the freezing bat cave, not freezing, 4 degrees Celsius bat caves, stuff like that, because of the cognitive function associated with humans. Yeah. So this is feasible for long duration space missions. If we could do synthetic hibernation, that would help with organ transplant and coma patients. So these animals don't get muscle atrophy. Yeah, they age much slower, and then ischemia reperfusion during surgeries, they can shunt blood to certain areas and not experience the detrimental effects. They also double their body weight and don't get diabetes, all of these really interesting things. So that is all I have. Pass it on.
Thank you, Anna. Really fascinating stuff you're working on, and I think it's interesting. I think this is a good opportunity to kind of see how this intersects with NASA's biological research. And so I'm going to ask Lisa Carnell to join the discussion. Lisa is the director of NASA's Biological and Physical Sciences Division. She oversees NASA's research on biological systems in space, including human health and uh, planetary protection. Welcome, Lisa.
So thank you. Thanks for the invitation to join the panel. This was so exciting. And I have to say, I just learned so much from Dr. Breit's presentation that I'll be reaching out to her for some really interesting touch points across my division. So I'm the director of NASA's Biological and Physical Sciences. And our division, we use the space flight environment, so mostly on the International Space Station, to really understand biological and physical systems. Biology behaves very different in microgravity, and so do physical systems. I'm sure you've seen a lot of the videos where the astronauts are trying to catch, a water bubble in their mouth. Fluids behave incredibly different, and so does combustion. Fire doesn't behave the same. So we do all of that work across my division. Our primary mission is to pioneer scientific discovery and then take all of that data that we learn from these early transformative studies to enable exploration and to address our agency's missions of sustainable presence on the moon and then moving humans out to Mars. And it's really great to me. One of the areas that I love is that everything we do is translatable right back here on Earth. And so you'll see some of that in the next couple slides that I'm going to show you. This is just an overview quick of the division and some of the things we've done. So interesting, my portfolio literally spans everything you just saw in that last highlights reel. That was just in 1 year of effort, the amount of work and research that we accomplish. And we have an incredible science community that takes all of that on, covering everything from microbes to quantum science. And one of the things, we did a really big shift in the past year because the administration has put out some emphasis on, sustainable presence on the moon. We just got a new administrator in December who has, basically given us the directive that he would like to start seeing more sustainable presence on the moon starting in 2028 and beyond. So we're working to make that happen, and then also looking for humans to become an interplanetary species. So how do we, really send humans safely out to the Mars environment and beyond? And one of those premises we have in our division is really to know before we go. And so we keep hammering that home. We need to understand what's going to happen to biology in deep space and uncover all those critical insights that we need to really ensure that we can bring the crew home healthy and safe. And we leverage all the different conditions in space on microgravity on the space station, sending things out to the LEO environment, and then deep space missions to understand that deep space radiation environment. So we like to say, and I'm in the Science Mission Directorate of NASA, that our science enables exploration, and that exploration enables our science. So coming up, I don't know how many of you have been watching the news, Artemis. Artemis is the next Apollo. So we have a mission coming up soon. We have Artemis 2 launching in early April. And I hope that you all are aware of this mission happening. If not, and you're hearing it for the first time, tune in. This is history in the making. This is our next big leap. And these are the 4 astronauts that are going to be traveling around the moon again. In the first week of April is our next launch window. It's about a 10-day mission. We have Jeremy Hansen on the left. He's a Canadian astronaut, Christina Cook, Victor Glover, and Reed Wiseman. And this crew is near and dear to my heart because they are an incredibly valuable part of our science mission that we have flying on board Artemis 2. So we've developed a science experiment that's flying on Artemis 2 to support astronaut health called AVATAR, which stands for Virtual Astronaut Tissue Analog Response. And these are, if you've heard of these unique devices, I was interested in seeing Dr. Breit's science that she was showing about the skin cells. So these are organ-on-chip devices, and they're seeding cells into microfluidic devices that literally mimic an organ system. So for example, if it's a lung model, it breathes like a lung or it beats like a heart. In this case, we created a bone marrow model, and each one of those astronauts you just saw has donated their cells to be part of this mission. And so we made a tissue chip out of each of them, and we'll fly alongside of them on Artemis 2. And this is going to allow us to really study and predict the effects of radiation on the individual. So we're going to be exposed to deep space radiation, which we can't mimic here on Earth well. And so that combination with deep space radiation and microgravity, which are 2 of the key stressors for the crew, we can now take a look at what happens in this environment. We chose bone marrow cells because they're very sensitive to radiation, and also that's where the immune system starts. So we're going to compare these samples directly to the crew themselves, and then compare it to pre and post mission data that we're obtaining. So really powerful study and getting us to that precision health where we can actually address the individual. And this is, as I was just mentioning, like we're paving the way for personalized medicine here with this. We're identifying their individual risks. And so this is a one-time, experiment. We plan to do many, many more of these to really build out and understand at the individual level. And hopefully, I think this would be great to translate this to, medical treatments here on Earth at the individual level. Not everybody responds the same to medication. I think we all know that well. And so how do we treat the individual? I mentioned to everybody, and I'd like to really emphasize this, that this was a huge undertaking. believe it or not, to get this experiment on board. Trust me, putting this into the Orion capsule with the crew was a feat itself. But pulling the science together with other agencies right here on Earth, and then small companies, and of course the Weiss Institute being involved in the organ chip itself. So really powerful science that I think really speaks well to the panel that we have here today. And just wanted to give a shout out that here's a QR code if you want to learn any more about the division, and you can follow us on X or Instagram. And that's it for me. Thank you.
Thank you, Lisa. Outstanding. Really interesting stuff as it relates to how humans deal with certain dangers when faced with harsh conditions. And so just really, really fascinating stuff. And I think that's an awesome segue to our next speaker, Amy Schmid, who is a professor of biology in Trinity College of Arts and Sciences. Amy's work centers on studying the life of extreme environments in extreme environments and astrobiology, focusing on how microbes survive conditions like those on other planets. Please welcome Amy.
Thanks so much for the invitation. I think I really appreciated hearing from Anna and Lisa on their work, and my hope is to connect together the relevance of my work in my lab with the search for life off Earth. I study life in extreme environments. You can see one of them behind me. And one of them on this title slide, it's at the Great Salt Lake, which is 10 times the salt saturation in the ocean. We would basically be pickled if we swam in it for too long. But a lot of times I like to introduce this topic is that most of the time we think about creatures that we see every day that we think can live through anything. You've probably found one of these in your house. And it's pretty hardy. It can withstand pretty dry conditions, cold, etc. Trees bend but don't break in the wind. They're nice and hardy. They live for hundreds of years. Some of you may be familiar with this cute little guy called a water bear or tardigrade. That's a small animal that can withstand years of complete desiccation without water. But I invite you to take a journey with me way beyond what we usually think about every day into life in extreme environments that are literally right beneath our noses. um, there's, like extreme life also elsewhere where we haven't been. For example, in Antarctica, they live in brine pools in freezing temperatures, below freezing, deep sea hydrothermal events under really extreme temperatures and mineral conditions. And the ones we study in my lab live in hypersaline ponds and lakes where they survive not only at saturated salt, but they transition flexibly between life within salt crystals being completely encapsulated and dried and life in the free water. And we like to understand in my lab using genetics and molecular biology techniques, the nuts and bolts of how it works. How do they live there? It's just absolutely fascinating to me. And in these lakes, like I said, it's feast or famine. So not only are they transitioning between salt and water, but they're also experiencing fluctuations in nutrient conditions and all kinds of things. And so one of the most remarkable features of these microbes is that when they are trapped in salt, they live within these little water bubbles or air bubbles, excuse me, within what are called inclusions within the salt. And geologists have found ancient salt that's almost 1 billion years old, and there's life inside. And so they find these cells that are surviving and alive 830 million years old. A lot of geologists are not microbiologists like I am, so they often stop and say, wow, it's alive. And then I say, but how? And so we're doing a lot of experiments in the lab to try to understand these types of stressors and nutrient swings and things like that in these hypersaline systems. And so we use a lot of live cell microscopy because we also don't know how they like to grow and live generally when they're actually happy. So we need to understand that first And this actually is really interesting because it goes well beyond what we just the extreme environments. It also teaches us about everyday cells and what they're resistant to, because the exception can often prove the rule. If we see something that's similar in our strange microbes to, say, human cells, that can teach us about general resilience that may be relevant to life in outer space. And speaking of outer space, this is a picture of the Great Salt Lake from the International Space Station. And the human impact on the lake is made visible even from space. So the north arm, where it's pink here in the image, is where we often go and sample. And so our research also takes us to these extreme environments where we can learn about how to preserve them. The Great Salt Lake is rapidly drying due to climate change and water diversion to agriculture. And so we're also actively engaged in policy and trying to save these extreme environments. This is just a bigger zoom in picture where we measure our samples. So salt lakes and ponds are a lot like Mars, especially in the crust where these microbes are living for hundreds of millions of years. They're dry, they're salty, they're irradiated. So here's an image from the other day from the Perseverance rover of the Martian environment. And as you can see on the salt crust on the edge of the Great Salt Lake in Utah is salty and very dry. And so there are these suspected salt crystals that came from drying at these different craters on Mars. And they've been shown to be as new as 1 billion years old. So 850 and 850 million and 1 billion aren't too far off. So if I had to guess, and if I were to look for life on Mars, this is exactly where I would look, in these sodium deposits. And I would not add water to them because these cells that I know in the salty environments, they explode when you pour water on them because they require the salt to maintain their osmobalance. And so what we learn in my lab is relevant for life, looking for life off Earth. And so what we've learned through my research is that we can understand where life can live, the limits of habitability, maybe in life on other planets such as Mars, as Dr. Carnell. I'm so excited about Artemis. And I'm hoping that we can also give some information from our research about protection of other planets, because knowing these extreme resilience features, we got to clean our spacecraft really well so that we don't contaminate where we land and risk mixing up the new life that we have to discover and the life we already know. So with that, I just want to thank our beautiful blue marble that we're trying to protect and preserve while we still go out and search. I'd like to thank my lab, especially my collaborator, Bonnie, from the Great Salt Lake, and funding sources. And I'd love to take any questions, and feel free to look up some of our research on Google Scholar if you're interested.
Very cool. Thank you, Amy. Thank you to each of our speakers for an outstanding presentation. And I want to now transition us to sort of a bit of a moderated Q&A, and then from there, we'll open it up to questions from the audience. So I'll start with question number one, and I'll start with Anna. So oftentimes in movies, we see astronauts sleeping in pods for years, and based on what you're learning from hibernating dwarf lemurs, what would be a realistic version of human hibernation for space travel?
Yeah, that's a good question. So there are different camps. I study thermoregulatory physiology, and I understand how expensive it is to send things to space. So the pods would be expensive, probably heavy. And also for maintaining cognitive function, I'm thinking, and a group of us are thinking that warm temperatures might be better. And that's why I'm excited about the tropical hibernation. So what I picture more is again, I don't know what temperature the spacecraft is going to be, but if you have a subset of the crew hibernating while the other drives or something, I'm thinking that a warmer temperature would probably be more realistic. So what I picture is humans able to hibernate at temperatures where the other crew members would be able to live their life. And if we could activate the genes, like we share so many of our genes with like our closest hibernating relative. So I picture just activating those genes and hibernating at 20 degrees Celsius. So no pods. So that's my opinion. But yeah.
Got it.
Mm.
Lisa, your work connects NASA with the NIH, the FDA, and private companies. Can you share your thoughts around any examples of space experiments or technology that are already influencing everyday healthcare on Earth?
Yeah, so as I mentioned, with the Avatar experiment, so interestingly enough, we actually joined forces with NIH, FDA, BARDA, several institutes in the NIH and CATS, NIAID, and NCI, and we funded about nearly $20 million worth of research a few years ago to advance these tissue chip, organ chip models. And their focus was, health here on Earth. How can we use these to really assess long effects after COVID, therapeutics that could be used to address COVID, like so the FDA was interested in toxicity from different chemical exposures. We're interested in space radiation and that. So I find it so fascinating because you can find intersections in some of the most unusual places. I had a lot of folks say, why are we working with NIH? We have incredible overlap. So everything that we're doing really translates to that human health impact here, but also accelerates our understanding for human health in space.
Wonderful. Amy, you study microbes that thrive in extreme environments. What can these extreme microbes teach us about how any cell, including ours, decides to whether to grow, repair, damage, or shut down under stress?
Yeah, that's a great question. So as I mentioned during my talk, a lot of times what we find is that the exception proves the rule. A lot of times everyone's like, oh, this must be really weird and it must have these crazy things that it does. And it actually has some weird things about it. So, for example, the inside of the cell is also saturated with salt as well as the outside. So they need to adapt all their molecules and everything. But then you also find things that are universal to life. And if you can find it in these far off reaches of the tree of life, it really teaches you about what the fundamental, really important mechanisms are for resilience within any kind of cell, any kind of organism. So, for example, they're the specialists at repairing their DNA. One of the ways that they can switch in and out of that salt and water, it sort of breaks apart DNA and chops it up into little bits. They're the experts in knitting their DNA back together. We can study those mechanisms and find those that are similar, but also maybe enhance, for example, make transgenic plants to enable them to withstand drought and so forth. So there are a lot of people, not my lab, but a lot of people doing applications of these kinds of resilience mechanisms. But I'm also really deeply interested in how all the relatedness of all of life and the evolutionary mechanisms involved. And so I think the exception can sometimes prove the rule.
Wonderful. Thank you. I'll get into maybe one or two more, and then I'll transition to audience questions. Um, Anna, from your experiment so far, what gives you the most optimism, and what are you most worried about as it relates to humans safely entering a hypometabolic state?
Yeah, this is a tricky one. If you would have asked me maybe 4 years ago at the last hibernation meeting that I was at, I was very weary of whether or not humans would be able to hibernate. Then we had another international meeting, and I'm more optimistic that it is possible. I think that again, it will have to be at warmer temperatures. Again, we see like a close relative able to do it. I think at least in my lifetime, it could happen like just a slow, like some reduction in metabolism, like smaller scale, maybe not for weeks at a time, but maybe for a 24-hour period. During my postdoc, we were working on looking at human cells at different temperatures, and thinking if we could start just a torpor bout, just a 24-hour drop in temperature, just in case an accident were to happen on the ISS, how long it takes, a couple weeks to get like a new emergency shipment up to the ISS. And so a small scale, could we just, save the oxygen, like lower the oxygen requirements, the food requirements just for a short period, before we could get an emergency response up. I think that is feasible. And once we get there, we can do more long-term stuff. The long-term, like months long, definitely like years long would be like, that doesn't make sense in my mind yet. Months long would be a huge jump. But I do think that it could be possible. But so that's my optimism slash worry, I guess, still. Yeah. And we don't have the cognitive response yet post hibernation, so the driving part is still up in the air. Our experiment just ended a couple weeks ago, so I'm excited to look at those data.
Thank you. I'm going to ask a couple of questions from the chat that I find that are really interesting. I'll start with you, Lisa. There's several NASA-related questions. So I'll start with this one. Can you share examples of some unexpected findings your team might have had about the human body and how it responds to microgravity and radiation?
Yeah, so we've had humans living on the space station for 25 years now, and had continuous human presence, and plan to have that continue on even after the ISS retires and we move on to new commercial platforms. We've learned even in the lower radiation environment, because believe it or not, going to the space station, the radiation environment's pretty low compared to once we get outside the Van Allen belts. So we have that protective magnetosphere that's really shielding Earth and everything that's in the low Earth orbit arena. And so that's why Artemis 2 is really important in that experiment. But what we've learned is, we get the astronauts have a higher risk of cataracts. So we know that this is a result of the radiation exposure. And so that's something that we've really been exploring and investigating. They have and it could be more adaptation responses as well, and could learn a lot from Anna and Amy in that area. But we've been watching what is adaptation and acclimation, and what do we need to push back to a normal state, Earth normal. And as they adapt, they still have extreme immune dysfunction. And so how do we understand that at the deeper level and adjust appropriately to make sure that they can stay healthy on these missions? And then we've also learned, we've seen some really interesting results in, in pressure changes. Obviously, I think that makes sense. It's pretty logical, right? When you're going to space and you have microgravity, you no longer have the same pull from gravity forces. So we see a lot of differences. We have a really critical problem called SANS. We nicknamed it, so space associated neuroocular syndrome. So you get a lot of pressure behind the eye, and we still are trying to understand what that is. So a lot that we need to learn there. So we've seen a lot. I could go on, but there are so many changes. And I do want to talk to Anna about some of the telomere dynamics that she's been seeing, because we also see some interesting telomere dynamics in the spaceflight environment that we were trying to make sense of. So much.
That's great. That's great. Just another quick, sort of a quick question: Is NASA sending any animals up to space during this next mission?
No animals other than the human. And I have to say, we're sending the human organ chip models because we're moving in that direction to be able to correlate directly to the human.
Very cool. This is to either of you. Can you describe a few basic medical procedures that are needed during extreme adaptation for zero or low gravity? Thinking of hypodermic needles and BP monitors, blood pressure monitors.
I can take that unless Amy or Anna want to jump in. And so we have, actually we invented we have invented several different technologies that to be able to use them in the spaceflight environment. We use ultrasound regularly in space, but that was, miniaturized so that we could use it for multiple types of disorders. And so ultrasound was one. If you're thinking about things that we need the ability to make saline and be able to do IV, capabilities. We developed a technology called IV Gen. It's kind of big, so it does work, but nothing works the same in space. So, even if you want to, put some sort of IV, insert that, you've got to have the right pressure to push the flow through because it's not going to be assisted with gravity. So every single thing that we do, whether it's feeding the crew, growing plants, watering things, everything requires some redesign to be able to adapt to the space environment.
Interesting. Lisa, is NASA a partner of Rocket Lab or SpaceX or a competitor?
Oh, are we a partner? Or oh no, we're partners. Yeah, no,
Yeah, okay.
we partner with all of these entities. We fund all of these entities. It's so exciting in the beginning, like seeing SpaceX take off. I think I can't remember what year was it, 2002, and it's been fantastic to watch them grow and then be able to be independent on their own. And Rocket Lab's doing amazing things. Blue Origin, we partner with all of these private companies and work with them. We have what's called Space Act agreements with many of them, and so they actually can work hand in hand with our experts at NASA to learn from us, and then so they can go on their own eventually. So it's fantastic, great partnerships.
Wonderful. Amy, um, based on the insights that you're developing from microbial survival and what you're hearing from your panelists, how do you think, this group could potentially work together in this regard, particularly on, human space research or Anna's work on hibernation?
Yeah. So like I said before, I think, the conserved principles of life in extreme environments are applicable. I think also the work that we do can really inform how we handle any space-based equipment and like any kind of,
Mm.
I'm really interested in interplanetary protection and avoiding cross-contamination because we have exactly one chance to find pristine life on another planet. And before we've already contaminated the moon, for example, and that was just based on, like in the 60s, we didn't know as much about the extreme life. So like a lot of microbes can still survive NASA clean room environments and things. And so I think it's, as humans start to explore farther and farther into the universe, I think it's really important to bear in mind to protect and clean those spaceships before we destroy any possibility of finding life elsewhere, because it's bound to be microbial and it's bound to be extremophilic. So I think
Yeah.
that can teach us about how to be good stewards of the universe, I suppose. Um, that's what I think about a lot.
Yeah, Lisa, I was wanting to have you maybe take a minute and go a little deeper on some of those invisible dangers that your team are exploring as it relates to sort of the longer missions on
Mhm.
planets like Mars, for example. Speak to maybe those dangers, and again, how are your team seeking to address those?
Yeah, so we know we've been measuring and evaluating the environment on the moon and Mars for
Mhm.
quite some time. We have a lot of data, but what we really don't have data on is human biology out in those environments and what would the effects be. And so I can tell you, and Amy, I don't know, you might cringe or get excited, but we actually are working. That's why I'm here today. We're working on a mission concept to send these human avatars, human biology out to Mars before we send the human being, so that we can understand what's happening in that deep space radiation during transit, which is a different environment. So we've got microgravity and 6 months of transit in deep space. But then once we land on Mars, we have partial gravity. Now we have different types of radiation. We'll get some albedo radiation coming off of Mars. It has a slight bit of atmosphere, maybe a tad bit of protection. So we really need to understand what happens to human biology before we send a human being. And to Amy's point, which I love, like this is such an incredible panel because I see so many touch points between Anna and Amy's work that's key in everything that we're doing. So Amy, you'll appreciate, front and center is planetary protection. So if we're going to send human biology out, like how are we going to ensure that we don't contaminate Mars, when we go? And we are completely all over that to make sure that, we're we're maintaining the right level of controls. Because we don't, like you said, we want to keep it pristine and know that if we discover life, that it wasn't something that we sent there ourselves. And so there are so many touch points and understanding that those radiation environments, number one, but then the partial gravity, because we've done so much in microgravity. When you get to the moon, you've got, a very small level of gravity, 6G. You get to Mars, it's a third G. So it's a completely different environment for the human body to adapt to. And we're not sure whether that will be beneficial or still have some negative results.
Okay, how close to reality are biospheres we see in fiction, such as The Martian?
I think Amy could take that one.
I have to admit, I haven't seen The Martian, and you all are going to like roll your eyes at me because the reason is I have a little bit of trouble with sometimes with sci-fi because it's not as accurate as it could be. But it tend like down to the molecular detail and things. But it really is dry and cold like that. I mean, and it's just like as dry and cold in the winter at the Great Salt Lake. So I mean, the environments I think are realistic, but like some of the scientific details could be. I mean, you can't do too many scientific details in a movie for entertainment, but that's my general opinion on science fiction. I hate to be a party pooper, but
I also don't like science fiction. It's crazy, I know.
I'm only laughing because we love it.
That's funny.
In
All right.
fact, Project Hail Mary is releasing this weekend, and I've got my tickets. My boss went to a premiere for it opening, so we're all excited. I have to say that reading those, I agree that they did a really good job for the most part because they did work with NASA scientists and folks at JPL and that to get as much of the science as accurate as possible. But we read it and like, oh yeah, there's some misses, but it's still entertaining.
yeah. I hear now that I hear from many people that I need to read it. I also see it in the chat. Okay, I will read it, folks, and I'll get back to you.
Yes. Yeah, read the book, as they say. That's great.
I
Fantastic.
think, I have to say, because in my division, we really focus a lot on how we're going to feed the crew as well. Like, so growing plants. So the Martian speaks to that, right? How are we going to grow crops off Earth and, sustain the crew in transit or on another planet? And so I got a lot of questions about the Martian. I don't know that potatoes are not going to be the first thing we grow, just an FYI.
Right. Very cool. Well, speaking, one more really quick question is, can AI help either of your research?
Yeah.
I
It
can.
sounds like the short answer is yes.
Yeah. Yeah. I
Yes.
mean, like we definitely, I mean, for us really quickly, we also take an approach of computational biology and we use we look at networks of molecules because it doesn't take just one molecule to survive in these extreme environments. And so far, we've used machine learning to infer the networks from large data sets. But I think AI is the next step in deep learning. And we use deep learning and protein structure prediction, for example, already. So yes.
Very cool. Well, I think we've come up on our time. Thank you, Anna, Amy, and Lisa, for a great session, and to our audience for joining us. Thank you, everyone.
Thank you.
Thank you.